Great. So now we'll open it up for lymphoma questions. Oh.
Yeah, so I had a question for both of you, and the indolent lymphomas and the aggressive lymphomas. Really just interested in the dosing, and if you could make some generalities of the dosing of bispecific antibodies that you're doing, how long you do it for, and is that related to how rapidly do you see rapid responses with these? And then you give a little extra. I saw one example where there was some maintenance bispecifics given. Just kind of wondering if there's any general strategies there, and kind of some follow-up questions on that, maybe related to, does everyone need IVIG? Does it just depend on if you're targeting CD19 versus CD20? Um, and how, if that's uniform or not.
Maybe I'll tackle the question about IVIG first, since that was just the last one. We've definitely seen high rates of infectious complications in our patients who are getting CD19 or CD20 directing products, so CAR T or bispecific. And so it's our practice here that if their immunoglobulin G level is under 400, then we do supplement them empirically. So I would recommend that. I think it's a little bit different in the myeloma world that you practice in, where there's, empiric IVIG that's given to your patients. So I'd say we're not quite there yet in terms of our program, but I definitely think supplementing if they're low is important. In terms of the rate of response or how quickly we see a response for bispecifics, I would say patients that we've seen, can achieve a very good partial remission or even a complete remission after 3 cycles of therapy. That's usually the first time that we scan them when they're put on a bispecific program. And so, I think that they can get to a CR very quickly when they're on this therapy. Your last question was, I think, about dosing.
How long do you do it in general? Are these being dosed for long periods of time beyond that period of response, or do we stop fixed duration? What do you do in that?
So at least in diffuse large B-cell lymphoma, the glofitamab trial originally, it was a time limited, with glofitamab. In the epcoritamab study, it was given indefinitely, but I think that you can see now that they're trying to revamp it and give EPCO in a more time-limited fashion. I think that's where the field will be moving.
I'm trying to address for indolent lymphoma. So currently, there's no bispecific approved by the FDA for marginal zone lymphoma, but we have 2 for follicular lymphoma in third line, Mosune and EPCO. The target dose has been established in the pivotal trials, and so the big question is the duration of treatment. And in a very arbitrary way, the pivotal trial were designed in a way where EPCO is given until progression or acceptable toxicity, and Mosune is given for either 7 or 18 cycles, 8 to 17 cycles. So after 8 cycles, if you achieve a CR, you can stop. If you have a PR, you go up to 17 cycles. But this was completely arbitrary. So to your question, that's the big question in the field, because of course, some patients do appreciate a fixed duration, and some others who have concerns that the lymphoma may relapse, they feel safer to stick to a continuous schedule. And so what we all think is that this needs to be MRD driven, because not all CR are the same, so we cannot just go by radiological response. The way historically we have measured minimal residual disease in FL, if you look into the big trials, has been qPCR, but we have very soon learned that it's not reliable, but in terms of sensitivity and specificity. We looked at, I think Dr. Camder actually looked at some point into clonoSEQ for FL and doesn't work well. Whereas, as you know, it's formally approved by the FDA for large B-cell lymphoma. CAPP-Seq performs a little bit better, but what I think is really gonna change the field, and it's currently incorporated in vast majority of FL trials, is PhasED-Seq, the MRD assay that I mentioned. So I do think that as we try to generate retrospective data from the pivotal trials of Mosune and EPCO and FL, and we learn after how many cycles is more likely to achieve CR MRD negative, that will inform duration of treatment. So currently it's completely arbitrary. To your second question, as Dr. Duarte said, so with bispecifics, this has been shown both for large B-cell lymphoma and FL. The median duration of B-cell aplasia in hypogammaglobulinemia is about 9 months from the last injection or infusion. And during that time frame, in addition to antiviral and anti-PJP prophylaxis, it's crucial to, in my opinion, also to follow IgG levels. and if they're below 400, to try to supplement with IVIG. In my personal experience, if you are very proactive on that, both on the antimicrobial prophylaxis and IVIG prophylaxis, I haven't seen much infection complications. And if they happen, it's usually due to lack of compliance from the patient part.
Okay, and maybe I'll ask one more question before Dr. Kamdar steals the mic. But I was wondering about the CELMoDs. This is kind of a niche question, maybe for BMS, but glucocorticoid I'd never heard of until today. But in myeloma, we have iberdomide and mezigdomide being developed. They all hit the same target at super potent levels. Why is this? Are those other drugs being developed in lymphomas and you just didn't talk about them, or is there something special about glucor and lymphoma?
Yeah, so I'll try to give a scientific clinical answer, though, maybe the rationale is probably the fact that LAN came off patent and we need a new glucocorticoid for Celgene BMS to make money. But so from a preclinical perspective, I share some preclinical data showing the way cereblon is targeted is a different pocket, and so the opening is supposed to be greater with golcadomide as compared to LAN. This should translate into better Aiolos and Icros degradation, and so the direct anti-tumoral activity should be better. So there's a hope that CR rates are going to be higher. As a matter of fact, if you compare, and I know we're always taught in medical school not to do inter-study comparison, but if you do, and you look into the CR rates with Golca plus R from the phase 1 that I'll show you today, and you compare to Augment trial, which is LAN plus R, with R-square, usually 20-30% of patients have a CR, and here at 0.4 was above 80%. So definitely it's hitting the target of being more potent. The other goal was to have a less toxic drug, and this is more related to how Golca indirectly activates T cells and NK cells. And as you saw from the adverse event table, we don't see the typical complications of LAN that I would say in my clinical practice of lymphoma. I wonder if it's the same in myeloma. I'm not familiar with the schedule in myeloma. It's mainly skin rash and fatigue. They don't seem to be a problem with Golca, but neutropenia is a big problem with Golca. And I know that in large B-cell lymphoma has meaningfully decelerated clinical development overall, particularly in combination with chemo. So I think that's still some work that needs to be done for Golca to be ready for a successful randomized phase 3 that's currently ongoing.
Thank you.
Thank you both for really excellent talks. Dr. Strati, one particular question, and this might be more of a philosophical question, but we know that follicular lymphoma is a very heterogeneous disease, right? There's a variety of different ways. Some patients can get relatively minimal therapy and have very durable remissions. And other people have more aggressive disease. We also know that the bispecific antibodies do carry a higher risk of infection, which the other questions have kind of addressed. I guess my question relates to, do you foresee modifications of risk stratification systems or other ways to sort of try to delineate a group of patients that might benefit from, maybe more delayed or later line bispecific therapy, not exposing them to the infectious risk up front?
Yeah.
Or how do you kind of think about that heterogeneity?
Yeah, so thank you for asking the philosophical question. And those are the questions that I like to address. So there are multiple layers of answer to this question. So the first one is we need to remind ourselves how we currently identify patients who do require treatment in FL. Because I mentioned at the beginning of my presentation, not every patient with FL, as you heard, needs treatment. About 40% of patients with FL, you can just watch over time and they will never require any treatment. And they may have a normal life expectancy, though I want to put a pin on that and tell you more in a second. So we do follow this criteria called GELF. So I think it's important to remind ourselves how GELF criteria were developed. These were completely arbitrary criteria where a bunch of French people met in a room 20 years ago for a large randomized phase 3 that was going on in France. And they, in completely arbitrary way, decided, more than 3 lymph nodes, more than 3 centimeters, one more than 7. There's no science whatsoever behind that. And I didn't present it today, but there was a very interesting oral presentation actually this year where the senior author was Dr. Falchi, presented by an MSK fellow, looking retrospectively into their real-world experience applying GELF criteria. And not all the criteria within GELF equally affect progression-free survival. And years ago, there was another retrospective study from Australia clearly showing that in clinical practice, many patients who meet GALACT criteria, they don't get any treatment, and many patients who don't meet GALACT criteria do get treatment. So there's a lot of heterogeneity. So I think that the first step that we have to take is trying to identify better criteria. And I think that they may not be clinical, and there's a lot of interest in trying to identify biomarkers that may help you, identify patients who truly require treatment subsequently, and then for those who do, those who may benefit more from immunotherapy as compared to chemo. And this is already happening. Right now, at this stage, there are very sophisticated type of analysis that would be impossible to integrate in clinical practice, like single-cell RNA sequencing, multiplex immunofluorescence, but hopefully, as we identify signatures of indication for treatment and signatures of treatment selection, then we will be able to also develop surrogate markers and sort of Hans algorithm for FL. The other point that I wanted to make is that we operate under the assumption that patients with FL have a normal life expectancy when compared to age and gender matched general population. But the retrospective experience on that is conflicting. Some studies do show that, like SEER registry data do show that, but other larger non-SEER based retrospective studies show that actually patients with FL, every relapse, they have a shorter PFS, and beyond the POD24, beyond the transform FL, they may actually experience a shorter overall survival. So I do think that it's still worth all the effort that we're making in trying to generate new treatment options that may be curative.
So I had a question for you, Dr. Stratney. Augment study excluded patients who were rituximab refractory. That was not the case with the EPCOR NHL study. I don't think the breakdown is clear how many were rituximab refractory who got randomized to R-square. My question is the rituximab required if someone is rituximab refractory in second line? And my second question is if, we understand if someone got treated with BR, then relapsed, POD or not, we can treat them with EPCO R-square. But if someone had rituximab and relapsed, do you feel comfortable going with the triplet? How do you extrapolate the rituximab refractoriness and prior treatments to implement EPCOR NHL as the new paradigm?
So that's, I knew that Dr. Cameron was going to ask a really difficult question. So I'll attempt. So the point that she's making is very important for ethical reasons, because in Augment trial, there was a large randomized phase 3 comparing R-square to rituximab plus placebo. Of course, you could not randomize patients who were rituximab refractory to rituximab again. It would have been unethical. But it's not the case in a trial where you're not randomizing to rituximab single agent. I think that, and again, this is a personal interpretation of data. I mean, if you look into the supplement data from at least EPCOR FL1, I don't think this was disclosed in mind. You do see that there was a good fraction of patients who were rituximab refractory, but the limitation is that not many patients had rituximab single agent before. So it's hard to extrapolate whether they were refractory to rituximab per se or R plus chemo. That's the first thing. The second thing is that what's the biological mechanism of resistance to rituximab. So the way rituximab works, it's actually mainly complement mediated as compared to obinutuzumab that engages NK cells and T cells. So most likely for patients who relapse, either don't respond at all to single agent rituximab, which are not many, or those who relapse within 1 year, 2 years, you name it, the biological mechanism of resistance is more relevant to the complement cascade. There's a very small fraction of patients who become CD20 negative, way more when used a bispecific. So I do think that rituximab refractoriness per se, unless the mechanism is a CD28 downregulation or negativity that we know is very relevant to EPCOR, should not limit the prescription on EPCO plus R-square in second line. And then, sorry, I forgot the second part of the question. Yeah. So you're saying like if somebody gets BR in front line, then of course, you would be comfortable using EPCOR square in second line. If somebody got rituximab in front line, again, my take would be as long as there is CD20 expression by immunohistochemistry or flow cytometry, I would be comfortable prescribing EPCOR square, understanding that some of the mechanisms relevant to complement cascade dysregulation may be addressed by the use of LAN. This has never really been fully investigated. But I really want to emphasize the issue with CD20 expression. So as a reminder, when you use rituximab based on the clone by flow cytometry that you are utilizing for CD20 expression evaluation, the negativity could be false. So it's always important for you to communicate with your pathologist and making sure that they're using the clone that does not interact with the by flow. It doesn't apply to immunohistochemistry by flow. And if the CD20 negative is true, I would definitely not utilize EPCOR square.
Okay, I have a question for Dr. Duarte. In these elderly patients with diffuse large B-cell lymphoma, how would you choose between our mini-CHOP and our POLA-GLOW?
Well, if we just go based off approvals, our POLA-GLOW is not yet approved, so we wouldn't be able to likely get it. I think that you could be creative and put them on POLA-R-CHP and maybe hold some of those agents and then say they've progressed and add GLOFI. But I think if we presume that they're both approved, I would be really hard pressed to pick an R-CHOP regimen in an elderly patient when the data behind POLA-R-GLOW is so good, especially if they have had prior cardiac complications or prior treatment for other cancers and maybe are limited in their anthracycline capabilities. To me, that data is just very impressive.
Thank you. And one last question for Dr. Strati. Now that 3 regimens are approved in second-line follicular lymphoma, like how do you choose between those 3?
Well, I don't have an answer, but I'll tell you what I think may happen in the near future. So first of all, I do think that we have to believe into randomized phase 3 trials. You know, we keep criticizing single-arm phase 2 that are currently increasingly bringing to accelerated FDA approvals. And then when we do get randomized phase 3, we still complain and we do challenge the results. So clearly, we should not use R-square single agent, R-square alone in second line, either TAF R-square or EPCOR square. So there are some, in my opinion, there will be scenarios where the decision will be made by biological factors. So let's say if in second line, the patient is CD20 negative, I will probably use TAF R-square. If it's CD19 negative, which is unlikely unless you're exposed to long or TAF or CAR-T, I probably use EPCOR square. It could be driven by logistics. The schedule for tafasitamab is not that friendly. And actually, the one for EPCO and EPCOR square is much better than EPCOR single agent. But it may depend on the ability of your patient to travel frequently to the infusion center, so they may make the decision for you. But I do think that, again, we're not supposed to do inter-study comparison. If you look at the curves for INMIND and EPCOR FEL1, the hazard ratio is much better than 0.2 for EPCOR square as compared to 0.4. The separation is visibly better. Of course, longer follow-up and rates of MRD negativity will tell us more. But personally, as of now, based on the data that I'm seeing right now, I would probably favor, just because of the higher chance for CR, EPCOR square, unless, as I said, biological factors or logistics make the decision for me.
Thank you very much. And so with that, we will have a lunch break. So enjoy your lunch, visit the exhibitors. The next session begins at 11:45. We'll have Dr. Sherbanow. Last but not least, great presentation coming up. Okay, thank you.